Okay, thank you so much everyone for coming. My name is Natalie. I run the customer support department at Diagnosys, and it is my very great pleasure to introduce to you Dr. James Akula. He is an assistant professor at Boston Children's Hospital in Boston, and he is a fantastic researcher. He's phenomenal to work with, and he's done some really exciting work on developing an optimized multifocal ERG method for rats with us. So without further ado, take it away, Jim.
Thanks, Natalie. So I just want to say that I am a user, not an employee of the company. I'm being paid to give this speech. I just like their stuff. And I thought that since the hot topic these days, of course, is artificial intelligence, that I would ask ChatGPT to get our juices flowing by giving us a nice poem about multifocal electroretinography in the rat. And of course it said yes, because it'll do anything. So, in research of the eye's inner sight, the multifocal electroretinogram shines bright. With light stimuli and a contact lens electrode, the retina's activity is revealed to decode. Rats are a model for human disease. Their eyes can be studied with great ease, using the mfERG to evaluate retinal function, to understand diseases and treatments with precision. Glaucoma, diabetes, macular degeneration, the mfERG can help with their evaluation, assessing the effects of drugs and gene therapy to improve outcomes with greater efficacy. But accuracy is key to the mfERG's success. High-quality procedures must be met with finesse to ensure the data gathered is reliable and true for the sake of science and the animal too. That's nice. So let us strive for excellence in our research. With careful technique, our knowledge will enrich, and in the end, we'll make great strides for the health of all, including furry guides. Now, I don't think that's actually a sonnet, right? Because the sonnet ends in a rhyming pair. So that's a little bit of an error, but not too bad for 10 seconds worth of work. Anyway, these slides here are produced by Diagnosys, by the company. This is a Celeris system, in case you haven't seen one before. It is basically a shoebox-sized thing with a lot of technology inside of it. I can tell you that you can record lots of ERGs very quickly if you want to, using simplified protocols. We don't do that. We record long protocols that take forever. But it's nice because you can customize it to give whatever protocols you would like. And I will also note that in addition to doing ERGs, we don't do many VEPs, but it can be used for that kind of stuff as well. And the one really convenient thing about it is the so-called touch-touch protocol, which is essentially if you're willing to record from only one eye or one eye at a time, you can use the fellow eye as the reference electrode. So that can be really nice. And as I said, you can customize the protocols, and if you want, you can do all kinds of things internally. You can put cursors on your A waves and your B waves. You can even process your responses in to extract the oscillatory potentials. I don't do that. I'm anal. I export the data as raw data and do everything myself, but that's just because it's the kind of person I am. Your mileage may vary. And that's not a real mouse, that's rubber. Here's a mouse being set up using the traditional full field stimulators. So these things have the electrodes and the light sources built into them, and that's really nice. You basically just plug them on each eye and go. This picture here is the thing we're going to be talking about today. This is the pattern stimulator. It's a little bit bigger, and the reason it's bigger is because inside that box there is a computer monitor, an OLED screen, on which they present the pattern stimulator. So those are the slides that are provided by the company. Now let me tell you my own stuff. So all of this was done in over maybe just 3 sessions over the past several weeks, playing around with really one of the first rat-sized stimulators that they've produced in my lab. And it was really fun just to sit there and see the kinds of responses that we can get out. Now, if you don't know anything about the ERG, you're probably in the wrong talk, but let me just tell you in case, right? The ERG is this measure of the electrical response of the eye to light stimuli, and is recorded at the cornea. And what makes the mfERG special is that the traditional ERG is a full field response, it's reflecting the activity of the entire retina, where the mfERG is a spatially resolved electroretinogram. And I think everybody can appreciate that while the mouse has mostly taken over as the model animal, the rat is still a valuable model for animal diseases, and it has a lot of similarities in its eye to the human eye. And therefore it is of great, I think, utility in having a device that can record electroretinograms, and specifically multifocal electroretinograms from the rat eye. Now, the mfERG has been used for millions of things in the human condition since it was first developed in the 1990s. Some of the things perhaps it's most famous for, but your mileage may vary, are detecting changes really early on in localized patches of retina that will then go on to develop disease, such as in diabetic retinopathy. It can be used now for studying gene therapy to regions of retina that are successfully transfected versus those that are not, to see what the changes are. And people can now use it because it is spatially resolved. We have other devices like optical coherence tomography, which came out around the same time, to actually try to co-register structural and functional information in patches of retina. So that can be really, really nice. In fact, we did a study on that. But I just want to point out, as a person who thinks a lot about eyes, in particular rat eyes, and how they are the same and how they're different from human eyes, these are two MR images that we've obtained in our group. On the left you see the human eye, on the right you see the rat eye, and they're really anatomically dramatically different, right? And one can, I think, just sort of appreciate, even without an understanding of optics, that you could see how a light could come into that human eye and be focused by that lens right on that retina and give a nice, beautiful, clear, sharp image. Does this make any sense? No, nothing like this could possibly be happening, right? So, it's not immediately obvious that you should be able to get any kind of resolution out of a rat eye with respect to visual acuity or multifocal electroretinography or anything. Nevertheless, there have been a few, and I apologize if I've missed any of the papers that do electroretinography in rats. So it's done, but there are really just a few. This might be an exhaustive list of all the mfERGs that have been attempted, and it's been about 3 years now since the first one was published. So that's because it's a royal pain in the butt to get mfERGs out of the rat, at least it was. So I have been working with the Celeris pattern stimulator for a long time now. I was originally working with it in mice. I believe the serial number of my PERG device is 5, so it's one of the very first ones ever made. And it worked nicely, but we've just recently gotten one that is sized appropriately for the rat eye. And I can confirm that while it does take a little more art than just a full field electrodes that you just sort of plop on and go, you do have to spend a little bit of time adjusting. Maybe it's a minute now instead of 10 seconds. It's not bad, and if you make a mistake, you just make a couple of quick adjustments and it's done. So 3 minutes for a challenging rat and 30 seconds for an easy rat or something like that. So with just a little extra work, you can reliably get these pattern stimuli to come nicely out of the animal eye using this really nice stimulator that they've developed. And so I have collected some data that I'd like to talk a little bit about today, but I'd just like to remind everybody, or tell you, in case you don't know, that the mfERG, just like the full field ERG, is totally non-invasive. It's recorded at the cornea using one electrode. It does not require multiple electrodes. And the way by which you can get these spatially resolved functional tests is by using a special set of stimuli that's governed by something called an M sequence. And an M sequence is just a mathematical sequence of ones and zeros that tell a hexagon typically, although it doesn't have to be a hex, when it should be on and when it should be off. And using some advanced correlative mathematics, you can figure out what kind of stimulus response is coming from what patch of retina. This is not a real mfERG. I just found a GIF on Google, but this is what the stimulus kind of looks like, although you would not see it repeat like you see it repeating here. And the M sequence is really advantageous over using truly general, or random, I should say, stimuli, because it has some very nice temporal properties that allow us to extract these things quickly, relatively speaking, so that's much better for the patient and for the rat. Now I want to start by showing you a traditional full field electroretinogram obtained in a rat. This is also from the Celeris system using those full field stimulators, and you can see that we can record responses in both the dark, where the rods are contributing, and in the light, where they have been silenced. And there's an obvious prominent negative going deflection in the dark adapted ERG, which we call the A wave, and that is known to originate in the rod photoreceptors. It is not present in the cone-mediated responses, but we do see a B wave in both of those cases, and that comes from the post-receptoral cells. Those are mostly bipolar cells. Okay? This is in contrast to a human full-field ERG. This is also obtained using Diagnosys equipment, but using their human system with a color dome. And here we again see an A wave, but now we have an A wave in both the dark and light adapted case. And that's simply because the human retina has a big chunk of the macula that is sort of cone dominated, whereas there's not very many cones anywhere in the rat retina. They don't give enough of a signal. So when you suppress the rod's activity out, you no longer get a photoreceptor signal. But we still see the same B wave. So the complement of post-receptor circuitry in the rat and the human retina is very, very, very similar. And that's important because the pattern ERG and the mfERG in particular is sort of a flickering ERG, right? It looks like every hex is kind of flickering, albeit at a random rate. And flicker can be too fast for the rods to track, but more than that, it just sort of light adapts the retina. So the rods are probably being mostly silenced in here. And so we aren't probably going to get much of a photoreceptor contribution. That being said, you might not think there should be zero rod contribution because in an M sequence, which is pseudorandom, sometimes we get a whole bunch of zeros in a row for a given hexagon, right? So there'll be an extended period of darkness there. And more than that, in every one, I believe, of those papers that I cited a few slides ago, they insert what's called filler frames. That is to say, black frames. They turn the entire stimulus array off for a few frames, and that's to increase the amplitude of the responses. And so it actually is possible maybe that you would see some negative going deflection early on originating in photoreceptors. This is a human mfERG, also obtained using a diagnosis system, the Espion, and it has a prominent negative going deflection, which sort of looks like an A wave, right? And then a positive going deflection following, which kind of looks like a B wave. And it's also got this second negative going deflection, which is sometimes referred to as the photopic negative response, or PHNR. Because when the mfERG was developed, we didn't know what the cellular origins of these things were. We didn't name them the same things we called them in the full field ERG, so we just call this N1 for the first negative going deflection, and P1 for the first positive going deflection, and 2 for the second negative going deflection. But we do now have some knowledge of where these different components of the mfERG come from, and the most elegant examples of that are actually pharmaceutical dissections of the ERG, where you can block synaptic transmission through various pathways in the retina using specialized drugs, and then do some math to add and subtract different response components and figure out, oh, this is coming from the cone-on cells, and the off cells, and the ganglion cells, and the photoreceptors themselves. Because it is spatially resolved, we can also do structure to function relationships, and so we did a mathematical exercise in the human retina. We can't inject humans with drugs. So our lines are actually those black traces, and the monkey pharmacological dissections are actually the red lines. I sort of superposed them, and the monkey mfERG and the human mfERG don't look perfectly the same anyway, but there's pretty good agreement there, I think. And what we can basically say is that yes, that initial negative going deflection is mediated by photoreceptors, and the P1 is probably mostly bipolar cells, and that inner retinal response, N2, or the PHNR, really is from the inner retina. So monkey work, and to a lesser extent our own statistical work, reveal that in some ways the mfERG really is just a mini regular full field ERG, I think. And while we don't know for sure that the same thing is happening in the rat, I would say we should probably assume it's true until we have reason to believe otherwise. That's what Akula would tell us. But I will also tell you that even just from the data that I'm going to show you today, we can get a little bit of evidence about where these things are coming from. If they're consistent with our hypotheses, that we shouldn't see much, for example, of a rod contribution in the ERG. So just some details. The stimuli today are covering probably 50 degrees of visual field. The whole monitor inside there is maybe 60 degrees, and they don't go quite to the edge. We can vary all kinds of dimensions, like the number of hexagons in our display, how bright the hexagons are when they're turned on, and how many of those blank frames we insert between stimuli. And obviously this is way too many dimensions to map out entirely, but we've done a little bit of work, just maybe 3 sessions, mapping out some of these things. So most of these tests are in N of 1 rat. Our rats are long Evans rats. Those are pigmented rats, which I think is important, and things might be a little different in an albino animal. Our anesthesia is ketamine xylazine, which is commonly used, although isoflurane is also popular. We do anesthetize the pupil with proparacaine. Perhaps, the cornea, perhaps you don't need to do that, but we do anyway, and we dilate the pupil using combination drops that are both cyclopentolate and phenylephrine, but you could probably use tropicamide or any drug of your choice. The active electrode is that pattern stimulator, and for our reference electrode, we do use the touch-touch setup, where we just use one of our full field electrodes as the reference in the other eye. And then we just clip a human ear electrode with some paste onto the tail. And after we position it, this is important, you do need to keep the cornea moist. Anyone who's worked with ketamine xylazine knows that opacification of the optical media is a major problem fairly quickly, but you can prevent that if you keep the cornea moist. And because there's a contact lens essentially built into the stimulators, as long as you keep that keep that eye wet, maybe every 20 minutes add one more drop, you can record for at least an hour and a half, and then the animals start waking up. So it's not because the quality of the imaging is poor. So here's a couple of rats set up in the device. You can see on the animal's left eye, on our right, there's a regular full field stimulator, and that big box attached to the animal's right eye, on our left, is the pattern stimulator. And here is one of the first mfERGs we recorded, 19 hexes. It's got 5 filler frames, I think, and I think it looks pretty good. Each of those colored bands on the right, that's an average of the rings, so the red one is just the centermost one by itself, and the yellowish one is all the 6 hexagons around that, and then the other one is the remaining one, is 11 hexagons around the outside, and they all look pretty good. One of the first things we did was add some blanks. That is to say, even when the M sequence had a 1 for that hexagon, it didn't turn the hexagon on. Keep it off no matter what the M sequence says to do. And I think there are 3 blanks in here. I'm hoping you can already guess which ones they are, but it's these 3 in case you need help, right? And so that works. That says that yes, we're not just recording scattered light or something across the whole retina. We really are getting a multifocal electroretinogram. So here's that 19 stimuli, hex stimuli again, now with the traces superimposed. As you can see, there isn't much of an initial negative deflection, and some of those there's not one at all, and they're in really good agreement. The rat retina is extremely homogeneous, for better or for worse. Now we move up to 37 hexagons. The responses have all gotten smaller, but they're still nice and clean, and I think they all look pretty good. And again, they're all roughly the same, regardless of eccentricity. Now that we move up to 61 hexagons, already I was sort of amazed that this could be done, but it comes out pretty nicely, I think. We are starting to see now some of the hexagons maybe dropping out a little bit, but if we look at ring averages, as shown on the right, they're very nice, they're all in agreement, and so that comes out quite well. By the way, 61 hexagons is something we do in the clinic sometimes. This is a commonly used clinical scenario. I would say the gold standard, or at least the default standard historically from back in the Veris days, was 103 hexagons, and so this is the classical mfERG, and it still works. Now we're starting to see definitely some areas that are dropping out. Maybe those are covering the optic nerve head, not sure, right? But we are now seeing very good, still eccentricity, similar responses. Dare we go beyond where we ever go in the humans, right? 169 hexagons. Well, individually now we start to see some of them we're looking for, but on average, except for that one red trace, which represents the middle hexagon all by itself, right? This is not an average of anything. It's still really good, actually. So yeah, we can go all the way to 169 hexagons, I think, and still get usable data. If you need that kind of resolution, then there it's available. So when I plot the summed amplitude, that is to say you take the average response and you divide it by the area over which the stimulus was covering, is that a slope on that line? I don't know, yes, maybe. So you might start to lose something as you increase the resolution, but certainly you can at least do 37 with great fidelity, and I think that's probably enough for most people. So it can do some pretty nice mfERGs even at relatively high resolutions. Now the next thing we did was we manipulated the intensity of the white flashes, right, when the hexagon was on, the one steps. And so here, I believe the dimmest one is around 27 candelas per meter squared, and the brightest one is just over 1,000 candelas per meter squared. And what we do is we just plot the response versus intensity relationship. I'm no longer normalizing by area, I just take the average response across the whole thing, because the number of hexagons doesn't change, so you don't need to normalize. And I now plot a standard logistic growth curve to those data. This curve has two key parameters. The first one is where the response is going to saturate, that's Vmax, and it looks like it's going to saturate somewhere around 3.5 microvolts. And then it also tells you how much light is required to produce a response at 50% that amplitude, and that's about 50. So that should be the dimmest light you want to use probably, is 50 candelas per meter squared. I recommend going higher than that. You probably want to go at least 4-fold higher than that, maybe even 8-fold higher than that. So 200, 400 candelas is probably good. No need to crank it all the way up, you get very diminishing returns thereafter. Now there's one really interesting thing. As I said, all the papers I believe that I cited earlier put filler frames in, and that's because undoubtedly mfERGs in the rat are smaller than those in the human, and they want to get bigger responses, so they inserted blank frames. Do you really have to do that? Every time you basically double the number of filler frames, you also double the length of the recording sequence, and that's exactly the same thing that you get when you add 1 bit to your M sequence, because it's a power of 2. So you can just trade off doubling the number of filler frames with decreasing your number of averages essentially by a factor of 2. So the question was, is it worth it? And so here we had 0 filler frames, that's essentially the human mfERG version, all the way up to 15 filler frames in between each stimulus presentation. And now I fit a slightly modified version of the function on the previous slide, and I can see where this curve is going to saturate, and it looks like it's saturating somewhere around 5 filler frames or so. You certainly don't need more than 5 filler frames. That's the most you ever want. After that, no more gain. And where does it semi-saturate? Well, around K1.5. That's not such useful information, I don't think. But so maybe 5 filler frames is where you want to go if you're trying to maximize your responses and still get your ERGs done in the shortest amount of time. But furthermore, what does the signal to noise look like? What do the shape of these curves look like? Here I have now normalized each of those responses by the amplitude of P1, essentially. And what we see is that that purple trace, which represents no filler frames, is actually very nice and maybe the nicest one up there. That is to say, though it was tiny, its signal to noise ratio is the best, and it has a really large photopic negative response. So if I just compare that to the human response I showed you a few slides ago, I think to me it looks the most like that response. They're all missing that initial deflection. N1 is not present because they don't have enough cones to make an N1. But they do make a photopic negative response that's quite nice. And if that's something of interest, you want to consider dropping your frames down and increasing the bits in your M sequence. So, in conclusion, the mfERG can now be conveniently recorded from the rat. We did all these tests in just a few sessions over a few days of hanging out in the lab, basically. I don't know exactly what the optimal pattern will be for your purposes. You have to decide what level of spatial resolution you need, but I'd say a good compromise for most would probably be somewhere around the 37 hexagon stimulus array. It's a nice average between compromise between plenty of spatial resolution as well as nice big robust responses. I would say you want to probably have at least 200 candelas per meter squared in your stimuli, maybe go up to 400 is probably better, but that should sort of be the low end of where you would consider based upon the intensity curve. But do note that even when we went down as dim as 25 candelas per meter squared, we still didn't see any rod intrusion. So if for some reason you want dim stimuli, but you're worried about the contribution of rod-mediated signals, you don't have to be worried. We don't get them even there. And depending on what you want, if you want more amplitude, you probably want to add some filler frames. And if you want to add filler frames, I would say stop at 5. That's about the most filler frames that you need. If you want to get the most human-like responses, don't bother with the filler frames. Just run your M sequence a little bit longer. Add a few more bits, and you can get a really nice result. So that's something you'll have to decide for yourself based upon your desired outcomes. But somewhere between 3 to 5 frames should certainly do it for growing those responses to larger amplitudes, more similar to those in the human mfERG. So Diagnosys has been working on these protocols. We're going to go back in the lab right after we get home from ARVO and maybe refine them a little bit, but I suspect that if you talk to them right now, they can give you some pretty good protocols to do your mfERG work. And if anything happens to make better versions of these protocols, I'm sure they're happy to send them out again. And I just want to add one more thing. This is not a protocol any of you will want because it's crazy and it's stupid, but it's fun. So we did go back to our 169 hex stimulus array and we added some blanks. We thought, what could we do? So we added some blanks. These are just randomly chosen. I don't know why we picked those ones, but we picked them just to see if we could really detect the blanked hexagons in a 169 hexagon array stimulus. And PowerPoint. There we go. So we colored them slightly differently. And now we block them out, and I really think we can now send each other coded messages, at least 3 characters long, using the mfERG in the rat. So it is pretty cool. It really does work. It actually resolves spatial function in a way that I would not have thought was possible before we started playing around with these animals, but it really does work nicely in the rat. So if you have any questions, please ask.